Okay, so now we are finally to the last step, the electron transport chain, and we're going to go through a process called oxidative phosphorylation. Now, looking at the picture here, I know this is kind of small, but look up here and notice here's our mitochondria. Here are our 4 steps: glycolysis, preparatory reaction, Krebs cycle, and finally our last step, electron transport chain or oxidative phosphorylation. I notice what we're doing here is we are taking hydrogens out of each of these 3 steps so we can carry the hydrogens over to the last step here. So what is a hydrogen made out of? Well, it's made out of a combination of an electron and a proton here. So when you see the H+, that's not the whole hydrogen atom, it's actually part of the hydrogen atom. And the NADH, that H does stand for the hydrogen atom though, and the FADH2, that does stand for the hydrogen atoms. So when we've gotten our hydrogens from the first 3 steps, and now here's where we're going to carry this to. Well, what are we carrying it to exactly anyway? So what we see here is that we actually see the inner membrane with the folds. So now looking at the inner membrane, the folds called cristae, here we have one of those folds. And notice we see our phospholipid bilayer. We see our matrix where this biochemical pathway took place and where we turned NAD plus to NADH and FAD into FADH2. And so notice here's our phospholipid membrane with different proteins embedded in that membrane, and something's obviously going on in these different proteins. Well, notice what we have here, the upper proteins, we have something called the electron transport chain. So obviously we're going to transport electrons through these proteins. So when the NADH brings the hydrogens here, the hydrogen kind of splits into 2 parts. It splits into an electron. Notice the electrons follow the red path, and what it actually does, it goes into this area that is a pump. Then we have a carrier that picks the electron up from that pump, carries it over to another pump. Then we have a carrier that picks the electron up from that pump, carries it to another pump here. Now, so the electrons are moving the pathway of the red arrow. We'll talk about what happens to the electrons at the end. But right now, just notice the electrons come here. This blue thing, this protein, picks up the electrons, carries it to another pump. Then this protein picks up the electrons, carries it to another pump. Now, if these are pumps, there's 1, 2, 3 areas here. What are they pumping? Well, they are pumping the other part of the hydrogen ion. They are pumping the proton. So we actually see a proton. When this electron gets here, it's kind of like electricity passing through something. It energizes, and that electron passing through here energizes the pump. I pump the proton from the matrix into this area, and this area right through here, called the inner membrane space, is actually the space in here between the inner membrane and the outer membrane. So I am building up a concentration of protons that came from the hydrogens that we carried here, and then we took the electrons out of it. We pumped the protons into this inner membrane space. Notice how I'm building up a concentration, and my electrons are moving through the system doing that. So in this case, I'm powering 1, 2, 3 pumps. Now, notice once the electrons move through here, then I've got to do something with those electrons. And what I'm doing with the electrons, here's where oxygen comes in. Here's the whole reason we breathe oxygen. I remember the first time I found this out, I was almost a little disappointed. But, you know, I like oxygen, and this is an important function, so it's okay. So what happens is oxygen comes in. Now, we know oxygen comes in as O2, but it splits into 2 individual oxygen molecules, so they aren't sharing that bond. So they each have 2 bonds open, and 2 bonds can accept 2 electrons from this, and then we actually have protons that are in here, and protons that are actually going to move back through this enzyme back in here. So if I'm oxygen and I can pick up 2 electrons and also pick up 2 protons, then I get converted from oxygen to water. So the oxygen that's here is going to pick up electrons and also protons, and of course that's an oxygen with 2 hydrogen atoms bonded to it through those polar covalent bonds we talked about earlier, and I form water. So oxygen I breathe in forms what we call metabolic water, which of course is inside the mitochondria. Of course, that's not enough to survive on. We still have to take in water from outside, but it does add to our water supply, and it does convert the oxygen that we breathe into something internal, which is the water. Now, but that still doesn't tell us how we get this energy here, because we know the majority of our ATP is going to be produced through this last process. So what else was happening? Well, I was bringing hydrogen from the NADH and bringing hydrogen from the FADH2. The hydrogen is breaking apart to my protons, which are being pumped into this inner membrane space. Again, the space between my two membranes. I'm building up a concentration of that hydrogen. There's hydrogen ions, or protons, which mean the same thing. And what's going to happen? Well, what happens anytime we get a high concentration of something compared to a lower concentration? Well, we talked about diffusion is where we move from high concentration back to a low concentration. And that's a passive transport. It doesn't require energy. It happens naturally. So what we have is something called chemiosmosis, where the protons that we pumped out now naturally want to move back into the matrix, as long as we have something here called ATP synthase complex, which means this is a protein, this is an enzymes. Now, keep in mind, what do enzymes do? Well, they either put stuff together, break stuff apart, or rearrange stuff by providing a docking place or a fit for their substrates, what they're putting together. Now, when ATP is used, remember we break off the N-phosphate group, and somehow if we need ATP again, what we've got to do is take that N-phosphate group that we broke off and add it back to the adenosine diphosphate so we can form ATP. Well, this is the enzyme that allows us to add the phosphate back to ADP, and that's why we call it phosphorylation. Oxidative, because oxygen's involved. Phosphorylation, because we need to add the phosphate back to ADP to form the ATP. Well, in order to do that, we kind of have to energize the enzyme for this action to take place. The movement of the protons back through the enzyme gives us the energy to cause the phosphate and to dock with the ADP. So now we can recatalyze that reaction and turn ADP plus a phosphate back into ATP. And then ATP can go through a protein channel, a facilitative channel that will then allow it to be reused for the many, many, many different things that cells do. Because most things cells do, if there's an activity, unless it's a passive transport activity, most things cells do require ATP, and usually a tremendous amount of ATP. So when we break it apart, we have to add it back together. And this is the process that does that. Now, if you kind of understand this process, we're going to go back and look at the numbers involved. But let me explain something before we do that. Notice when NADH enters this process, notice it's energizing 1, 2, 3 pumps. When FADH2 enters this process, it never enters has anything to do with the first pump. It goes to the carrier and it only energizes 2 pumps. So the key to understanding the amount of ATP we use is to understand the difference between whether we energize 3 pumps, 1, 2, 3, or whether we energize only 2 pumps, 1, 2. If we start here, we're going to only energize 2 pumps, 1, 2. But if we start here in this electron transport chain, which we find in all these cristae, then we can energize 3 pumps. So remember our numbers, 2 and 3, because that's going to become important for what we're going to talk about next.